In controlled environment agriculture or indoor agriculture, the spectrum of lighting can regulate a wide range of traits. Blue light generally suppresses plant growth but promotes anthocyanin production to intensify leaf color. Green light does not change most crop traits, but sometimes can reduce plant growth and color. Red light excels in photosynthesis to promote plant growth, but does little in secondary metabolism to enhance leaf color. Far red light drives photosynthesis effectively and brings about shade avoidance responses to increase plant size, but it reduces leaf pigmentation. In artificial lighting, these individual wavebands are often combined at various ratios to keep a balance of desired plant attributes. In the majority of indoor farms, the light spectrum is static, which means it is the same throughout the entire crop life cycle. From seed to harvest, lettuce goes through three phases: the lag phase of seedlings, the exponential growth phase of maturing plants, and the finish phase before harvest. The desired crop attributes are somewhat different in the three phases. Plants should be easy to transplant during the lag phase, grow fast during the exponential growth phase, and achieve high yield, great color, and high nutritional value at harvest. A single spectrum like 10% blue plus 90% red is fine to grow lettuce with, but not enough to check all of the boxes in all phases. With an understanding of spectral effects, we can use this spectrum during the lag phase, but change the spectrum later on by using LEDs with adjustable spectra, or by moving plants to different spectrum zones. For example, in the exponential growth phase, we can add far red to boost growth. In the finish phase, we can increase the ratio of blue to red light to increase pigments and nutritional compounds. Even though high blue light slows down growth, since it is applied for just a few days, we can still achieve pretty high yield at harvest. If a visually comfortable environment is important, we can mix in some green light to create white light, which makes it easier to inspect crops. In this study, we focused on spectral variations during the lag phase and the exponential growth phase. There was no dedicated finish phase. We wanted to see how changing the spectrum during the lag phase or the exponential growth phase influenced lettuce growth and color at transplant and harvest. We postulated spectral effects in the first phase would persist through the second phase, regardless of the subsequent spectrum. However, spectra applied in the second phase would have a larger impact on final crop traits than the first phase. Also, we wanted to determine whether changing the spectrum offered any practical benefits, and which spectral combinations were ideal for the two phases. We grew red leaf lettuce rosé in a climate-controlled room under a 20-hour photo period. The air temperature was set at 23 degrees Celsius. We propagated seedlings in rockwool cubes. On day 11, we transplanted them into a deep water hydroponic system on 3-tier racks. The nutrient solution had an average pH of 6.1 and EC of 1.8 millisiemens per centimeter at a nitrogen concentration of 125 to 150 ppm. CO2 was at the ambient level. We created 6 spectral treatments with the same total photon flux density of 180 micromoles per square meter per second. They were warm white, red, blue plus red, blue plus green plus red, blue plus red plus far red, and blue. The subscript here after each waveband shows its photon flux density in micromoles per square meter per second. Warm white LEDs produced blue, green, red, and far red photons. Alongside the blue plus red treatment, we partially substituted 60 micromoles of green or far red for red. We delivered each spectral treatment at a random canopy in a randomized complete block design with 2 blocks. This image shows 6 treatments in one of the 2 blocks. Data were pulled from 2 blocks. This is lettuce grown under static treatments for 11 and 25 days. The differences among treatments were mostly consistent on day 11 and day 25. At a glance, we see longer, greener leaves under warm white, red, and blue plus red plus far red. In contrast, plants were smaller and redder under the other treatments. These observations were confirmed by data. In these charts, means followed by different letters within each parameter are significantly different. Trends were similar between fresh mass in light green bars and dry mass in yellow dots. Unless far red was included, blue light decreased biomass. Conversely, biomass was highest under treatments with far red or no blue. Compared to blue plus red, adding green light did not influence biomass on day 11, but decreased it on day 25. This indicates effects of green light can change with the plant age. On the other hand, adding far red to blue plus red increased fresh mass, but not dry mass. Leaf length is an indicator of extension growth. Leaves were longest under treatments with far red or no blue on day 11, and were longest with high far red on day 25. Although 100% blue produced the shortest leaves on day 11, it promoted leaf elongation on day 25 like warm white and red. Adding green light to blue plus red increased leaf length on day 11, but did not change it on day 25. In these cases, spectral effects changed with the plant age. Now that we have explored static lighting, what happens if we expose plants to different spectra in the lag phase and the exponential growth phase? On day 11, we transferred plants under one treatment to every treatment. For example, if we have 6 plants under warm white, we will take each plant to each of the 6 treatments and continue to grow them until day 25. Each plant here represents 8 subsamples. We did the same transfer for plants under the other 5 treatments: red, blue plus red, blue plus green plus red, blue plus red plus far red, and blue. We took time-lapse videos under blue plus red, with and without green or far red, to track real-time growth patterns. Every second of time-lapse playback shows 5 hours of real life. Night periods were cut out. Faster leaf expansion under green or far red was evident early on. On day 11, treatments transfers took place. Plants continued to grow. Now, plants looked different under the same treatment because some were transferred from the other treatments. As plants matured, they started to look more different between treatments than within each treatment. Finally, the plant canopy closed first under blue plus red plus far red. We evaluated spectral effects on final lettuce traits based on application time. For example, to determine effects of warm white applied during the lag phase, data of all plants grown under warm white for the first 11 days were combined for analysis, regardless of the second phase spectrum. Similarly, to determine effects of warm white applied during the second phase, data of all plants grown under warm white from day 11 to day 25 were combined for analysis, regardless of the lag phase spectrum. Even though plants were small during the first 11 days, the spectra they received had a lasting or residual impact on final lattice traits like biomass. In comparison, spectra applied on days 11 to 25 had a more pronounced impact, evidenced by larger variations among treatments. Simply put, every experience counted, but the more recent experience carried more weight. The trends here were similar to those of static lighting. Now, if we want to have the highest yield on day 25, we can identify the most productive spectra in two phases. In phase 1, they were warm white, red, or blue plus red plus far red. In phase 2, it was red. Of course, we can use a static red spectrum throughout to achieve the highest yield. But the thin, tall, and delicate seedlings need to be handled with extra care during transplant, which can increase transplant time and labor cost. If 10% blue light is included in the lag phase, compact and robust seedlings are easier to transplant, although there is a 9% reduction in final fresh mass. However, this yield reduction may be diminished if plants grow for another week or two to reach marketable yield. Final leaf length and color were mainly controlled by spectra applied in phase 2 instead of phase 1. The trend of leaf length in phase 2 was similar to that of static lighting in the same phase. We quantified leaf greenness and redness with a colorimeter. The lower A star is, the greener the leaf. The higher A star is, the redder the leaf. Since anthocyanin production can be rapidly increased within just a few days in a blue-rich environment, it's no surprise leaves were reddest under 100% blue applied in phase 2. Adding green or far red to blue plus red in phase 2 decreased leaf redness. In conclusion, spectral effects in the lag phase had a lasting impact on final lattice traits, although those in the exponential growth phase were more pronounced. Changing the spectrum in two phases did not increase yield compared to static red light, but it helped achieve desired traits in each phase, increasing flexibility and precision of control. Ultimately, spectrum selection in each phase depends on a grower's goals, considering factors like desired yield, morphology, and color, as well as logistics, cost, and fixture efficacy, among others.